In this lecture, we're going to look at cellular respiration in a little bit more detail. We're going to look at the major events of each major stage of cellular respiration. We're not going to get into every little detail. What I like to do in this lecture is provide a framework that you can plug details in later. If you take a more advanced biology course, a majors level course at some point in time, you might be expected to learn a lot more detail about cell respiration. But I would like to lay this framework of information in your brain so that later you have something to plug those details into. So we're going to just talk about the major events for each stage of cell respiration. And when I say stages of cell respiration, what I'm referring to is there are going to be 4 major stages we are going to talk about. The first is going to be glycolysis, which happens out here in the cytoplasm of the cell. So when glucose enters the cell, the first thing that's going to happen out here in the cytoplasm is a process called glycolysis. We're going to take that 6-carbon molecule and break it down into 2 3-carbon molecules. We're also going to generate some ATP when we do that. And then if oxygen is present, we're going to move into the mitochondrion and we're going to perform more cell respiration and generate more ATP. So we're going to go through this stage called pyruvate oxidation, which isn't written on this screen. And then we're going to go through what's called the citric acid cycle, where we complete the breakdown of glucose. And then at the end, we're going to go through oxidative phosphorylation, which is going to include the electron transport chain and chemiosmosis. This is where we're going to make most of our ATP. So remember the general formula for cellular respiration. It's glucose plus oxygen, and then we end up with carbon dioxide and water as our products. But we also make 36 to 38 ATP on average. For this lecture, we're just going to use the number 36 so that we can keep track along the way of how many are generated in each stage on average. We're just going to use 36 as our number. Remember that this is a biochemical pathway, which means this arrow does not represent one step. It's going to take as many steps to get to these products. So this arrow actually represents multiple steps to get here. If we released all of the potential energy in this glucose molecule all at once, we would burn up our cells. We need to release this energy a little bit at a time and then package it as ATP to use anytime, any place. This is a great plan because it's not like fueling up your car where you have to have a constant supply of fuel in order to produce energy. In this case, we can consume some fuel and then we can package it as this energy currency that we can use anytime, any place in the cell. That is pretty amazing. So we're going to look at glucose. At the end, I'll show you just briefly what happens with fats and proteins, but we're going to look at what happens with the carbohydrates that we eat. This is the preferred source of fuel for the cell. So if you're on a low-carb diet, you are not giving your cell what it needs to perform this very, very, very important reaction. Probably the most important chemical reaction that happens in your body. So let's start looking at the details. There are a couple of terms you need to understand. The first term you need to understand is what is an electron carrier. We're going to have 2 electron carriers that are part of the cell respiration story. NAD, it's actually NAD+, and FADH. These electron carriers are going to pick up electrons in the form of hydrogen. Those electrons are carried by hydrogen. Remember, hydrogen is atomic number 1. It has 1 proton and 1 electron. We have 12 hydrogens we're going to need to pick up as we go through this story. And those hydrogens are going to be picked up by these electron carriers. So electron carriers are important to understand. Here's just a quick picture of nicotinamide, NAD+. You don't need to understand it in great detail. You just need to understand that this is what's going to shuttle those hydrogens through this process. Okay, so back to the overview. So we're going to go through a little bit more detail for each stage. We're going to talk about how many ATP are generated at each stage, what we start with and what we end up with. We're going to look at where we generate these 6 carbon dioxides, how and when we make the water, and how we complete this breakdown of glucose to generate 36 ATP on average. Okay, so let's start going through the details. We're going to start with glycolysis out here in the cytoplasm of the cell. So first stage of cell respiration, glycolysis. Glycolysis, this means sugar splitting. We're going to start with a 6-carbon glucose, and we're going to go through the steps of glycolysis, and at the end we're going to end up with 2 3-carbon molecules called pyruvate. Let's look at this in a little bit more detail. Again, you don't need to know the details of this. You need to know a couple of details, but not every step of glycolysis. So glucose enters the cell, and out here in the cytoplasm of the cell, we're going to go through these steps of glycolysis. Glycolysis actually requires 2 ATP. We need to get this process started. Remember, we talked about activation energy. Even if that boulder's at the top of the hill, you need to give it a push to start going. This is an exergonic reaction. It's going to release energy, but we need to get it started. It requires 2 ATP to get started, but we're going to make, I'm going to put make in quotes, we're going to make 4 ATP. So we end up with 2 ATP net being generated through glycolysis. I talked in the last lecture about energy and how we don't really make ATP. We don't start from scratch and put all these atoms together to make ATP. Instead, what we're doing is rephosphorylating ADP. Remember, ATP in its cartoon form is adenosine triphosphate. The last bond is shown as a squiggly line because it's a high energy bond. The way ATP powers reactions is it transfers this phosphate group to another molecule, and when it does that, it becomes ADP, adenosine diphosphate. What we're doing in cell respiration is we are rephosphorylating ADP to become ATP again. We're going to put that last phosphate group back onto ADP to regenerate ATP. That's what cell respiration is going to do. So when we say we're making ATP, we're not making it from scratch. We're really just rephosphorylating ADP. In glycolysis, the way we rephosphorylate ADP is using an enzyme. And because we're using an enzyme to transfer that phosphate group back to ATP, we're transferring it to ADP to form ATP again. It's called substrate level phosphorylation. Remember, the substrate is the reactant that binds to the active site of an enzyme. And because we're taking this phosphate group from a substrate and transferring it to ADP, we call that substrate level phosphorylation. So we make these 2 ATP in glycolysis by a process called substrate level phosphorylation. If you don't remember what phosphorylation means, you should rewatch the energy video. Recall that phosphorylation is a general term which means transfer of a phosphate group from one molecule to another. In this case, transfer of a phosphate group from this substrate to ADP to regenerate ATP again. So in glycolysis, we make 4 ATP via substrate level phosphorylation. It ends up being a net of 2 because we use 2 to get this process started. So that's one of the very important events of glycolysis, is we make 2 ATP from that glucose. Another important thing we do is we start picking up those 12 hydrogens. We're going to pick up 2 of those hydrogens using our electron carrier NAD+. And when NAD+ picks up that hydrogen, it becomes NADH. Okay, so in glycolysis, we are going to generate 2 ATP via substrate level phosphorylation. We're also going to make 2 NADH. We're picking up the first 2 of our hydrogens from that glucose molecule. And at the end of glycolysis, we have taken that 6-carbon glucose molecule. We've gone through all of these steps, and we end up with 2 3-carbon molecules called pyruvate. So we start glycolysis with glucose, we go through all of the steps, we end up with a net of 2 ATP by substrate level phosphorylation. We pick up the first 2 hydrogens, so we generate 2 NADH, and we end up with 2 pyruvate as our end products. We do not generate any carbon dioxide or water during this stage of cell respiration. So these are the details. This is our energy investment phase, and then we have our energy payoff phase, where we end up with 2 pyruvate. So in glycolysis, we make 2 ATP net by substrate level phosphorylation, 2 NADH, and we end up with 2 3-carbon pyruvate molecules. We don't generate any of our carbon dioxide yet. Let's look at the details. Again, you don't need to know each step of glycolysis. It's actually even more detailed than this. You can see we use an ATP here, we use an ATP here. We generate 2 here and we generate 2 here. You can also see that there's an enzyme used at each stage. Pyruvate kinase. Ends in ASE, so we know it's an enzyme. So if you want to see the details of glycolysis, you can. You can look at your textbook and see the details of glycolysis. What you need to know, though, is at the end, we end up with 2 ATP net. We picked up 2 of our hydrogens, and they're being carried through the process, and we made 2 pyruvate. Okay, we're also going to generate some water during the process, but we do not make carbon dioxide yet. We're not going to track waters as we go through this process. You have enough detail to remember without tracking the waters. These are the major events of glycolysis that you should know. Okay, we end up with 2 pyruvate. Now, if oxygen is present, we're going to keep going. Glycolysis is the only process that can happen with or without oxygen. If oxygen is present, it's called aerobic respiration. So aerobic means O2 is present. If O2 is not present, it's called anaerobic respiration. So an means without. Anaerobic respiration. If conditions are anaerobic, it means there's not enough oxygen present. No oxygen. Glycolysis can happen with or without oxygen. If oxygen is present, then those 2 pyruvate are going to enter a mitochondrion, and we're going to continue the breakdown of glucose. And we're going to continue generating ATP. If oxygen is not present, I wrote with or with. This should be with or without. If oxygen is not present, then we're going to go through fermentation. And we'll talk about fermentation after we go through the steps of cell respiration. Okay, so these two pyruvate, if oxygen is present, we're going to go through the second stage of cell respiration. And that stage is called pyruvate oxidation. So if oxygen is present, we're going to go into a mitochondrion and we're going to go through the second stage called pyruvate oxidation. Why do we need to go through pyruvate oxidation? Because pyruvate cannot enter that citric acid cycle, which is where we're going to generate some more ATP and keep the process going. Citric acid cycle is where we're going to complete the breakdown of glucose. But pyruvate can't enter the citric acid cycle. We need to convert it to something called acetyl-CoA. So this is the details of pyruvate oxidation. We have two pyruvates that we generated, so each one is going to go through this process. So we enter the mitochondrion. It's going to go through a transport protein. We're going to generate, for each pyruvate, we're going to generate one CO2 and produce one acetyl-CoA. But we have two pyruvates. Remember, we took that 6-carbon molecule and split it into two 3-carbon pyruvates. So those two pyruvates are going to generate the first two of our carbon dioxides. And you can see what's happening is we're shaving off this carbon and these two oxygens. So we're ending up now with a 2-carbon molecule called acetyl-CoA. Co is standing for coenzyme. We also, for each pyruvate, generate another NADH. So a total of 2 carbon dioxide, 2 acetyl-CoA, and 2 NADH. Now this acetyl-CoA can enter the next stage. The next stage of cell respiration is called the citric acid cycle. It's also called the Krebs cycle. Krebs is the guy who discovered this process. Again, you don't need to know all the details of the citric acid cycle. Here are the details. You do not need to know this level of detail. You need to know we start with two 2-carbon molecules called acetyl-CoA. And each one of those is going to go through this. So for the one glucose we started with, we're going to go through this twice because we have two acetyl-CoA that enter the cycle. You can see for each acetyl-CoA, we're going to make 1, 2, 3 NADH and 1 FADH2. And we're also going to generate 1 ATP. What's significant though is we complete the breakdown of glucose. When we finish the cycle, there is nothing left of glucose. It is completely gone. We have released it all as carbon dioxide and we have picked up those hydrogens. So in the citric acid cycle, we start with 2 acetyl-CoA. We're going to generate 2 ATP. For each one, we're going to make 3 NADH, so that's 6 NADH total. And for each one, we make 1 FADH2, so that's 2 FADH2 total. And you can also see that we release 2 carbon dioxides per acetyl-CoA, so that's 4 total. How do we make these ATP? We make these ATP, again, by substrate level phosphorylation. Substrate level phosphorylation. using an enzyme and a substrate to transfer a phosphate group to ADP to make ATP. I'm going to just abbreviate that SLP, substrate level phosphorylation. Substrate level phosphorylation, the same way we generated ATP in glycolysis. So what do we have left at the end of citric acid cycle? Well, glucose is completely gone. So this completes the breakdown of glucose. But how many ATP have we made so far? We've only made 4 net. Remember, we made 2 net in glycolysis. Now we've made 2 more, so we've only made 4 of our 36. We have 32 more ATP to go, but glucose is gone. So where is all of the energy from that original glucose molecule? Well, guess what? It's in the hydrogens. It's in those electron carriers. And this is the unbelievable part of the story to me. So let's count. 2 plus 6, that's 8. 10, 12. That's our 12 hydrogens. So we picked up 2 in glycolysis, 2 in pyruvate oxidation, and the other 8 in the citric acid cycle. Now all those hydrogens are going to be a huge source of potential energy. As those go through the process, they're going to hit what's called the electron transport chain, and that's going to generate the rest of our ATP. That's going to be a very important part of the story. But before we get to that part of the story, I just want to do a quick summary of what happened at the first 3 stages.